Okay, cool. Yeah, okay. Okay. So thank you, Irina, for the introduction and all the people that are attending this seminar. I will present the project that we entitled Time Resolved X-ray Solution Scattering Unveils the event leading to hemoglobin in capture by staphylococcal IsdB. And indeed, yes, so sorry, this slide is gone. Indeed, our biological system, the system that we are studying is about the finding of new antimicrobial target to defeat antibiotic resistance. In fact, it is expected that by 2050, the death caused by antibiotic-resistant bacteria will be like 10 million, the same as the current cancer mortality. And we focus on Staphylococcus aureus, which is a WHO high priority pathogen, and that is also a part of the ESCAPE group that has a list of most elusive bacteria to antibiotic. And aiming to find new target, we focus on the iron acquisition from Staphylococcus aureus, which is a virulence-related mechanism. Indeed, during starvation, which is a common condition when the bacterium is infecting human, it expresses the iron regulator surface determinant ISD system to obtain the hemic iron, which is bound to hemoglobin. In this picture, we can see how Staphylococcus produces hemolysins that breaks the red blood cells up and led to release of hemoglobin in the bloodstream. This is the source of iron for Staphylococcus aureus, and if we focus again, we can describe how IsdB, one of the protein from the ISD system and our protein of interest, is binding hemoglobin and then extracting the heme. The heme that is extracted is then transferred inside the cell by other ISD protein to release iron inside the cell. All the protein of the ISD system are also called hemophore. In fact, they are binding or interacting with heme in some way. Interestingly, IsdB mutant strains are severely impaired in the ability to grow when they are cultured with hemoglobin as a sole iron source. So, zoom in again. We have two different steps that we can characterize, and still we have already published a paper in 2019 where we've been able to follow the binding kinetics of IsdB to hemoglobin by SPR, and also the in transfer from hemoglobin to IsdB by visible spectroscopy. Indeed, hemoglobin has a very distinctive pattern of peaks in the visible range with a high intense peak at 400 at 6 nanometer. This pattern changed a lot when IsdB is added, so when the in transfer is finished. We have a new visible spectrum that is the one that depicts the extracted heme. And if we follow the signal at 406 nanometer over time, we can also characterize kinetically this process. What we found but never published is that we can also use the intrinsic tryptophan fluorescence of IsdB to characterize its binding to hemoglobin. Indeed, if we add hemoglobin to IsdB, the fluorescence emission of the hemophore, so the bacterial protein, decreasing a lot. and this is the emission, the fluorescence of the complex. So we have another technique that we can use, but none of them can give us any insight under a structural point of view. So for this reason, we decided to use time-resolved wide-angle X-ray scattering to characterize the dynamic interaction between IsdB and hemoglobin under a structural point of view. A quick recap, just for many of you that maybe is not familiar, but I don't know if there's some. If we have protein crystal, we can just irradiate them with X-ray and obtain diffraction. While if we have protein in solution, we can still irradiate the sample, but we will have a scattering pattern. And if we look at this picture, um, we can say we can see that X-ray crystallography, so having crystal, lead us to a very high spatial resolution, while if we use wax and SUX, that are solution X-ray scattering technique, the resolution is very limited. Under a time resolution point of view, we can have different techniques. The direct laser excitation that has a very high temporal resolution. Then we have laser TJAM and rapid mixing. So unfortunately, our protein interaction cannot be started by direct laser excitation, nor by laser TJAM. So we went for rapid mixing, as we will see in the following slide. But before, I want to jump back to the biological question and see how our protein complex can be complex. So if you might know, the hemoglobin is usually for tetramer, which are a dimer of alpha beta heterodimer, while IsdB is a single monomer. So in the biggest complex possible, we will have an hemoglobin tetramer bound by 4 IsdB molecule, but this was already been proven by other researchers that is not possible because of steric clash, and also these two complex with 3 hemo4 bound to hemoglobin are not possible. So the first possible complex are the one formed by hemoglobin tetramer and 2 hemophore, either bound to an alpha chain and beta chain, both bound to the alpha chain, or both bound to the beta chain. And then we can just scale down and go with one IsdB molecule bound to an hemoglobin tetramer. And also if hemoglobin will dimerize, we will have also these complexes. And if the hemophore binding is causing a big stress to the human protein, it will also monomerize, leading to the formation of these small complexes. Moreover, we have to remember that the binding of IsdB will also go lead to the heme extraction, which will further stress under a structural point of view hemoglobin. So we will have a lot of possible oligomeric species in solution, and this is not good for solution X-ray scattering because we only have, as a result of this technique, a solution, a scattering pattern, which is the average of all the scattering of all the protein in solution. And if they are heterogeneous, we will probably have an average signal that is not informative. Unfortunately, the molecular weight of all these species is not that different, so it is very difficult to separate them by size exclusion chromatography. So before going for time-resolved wax, which is a very promising technique, but also can be sensitive to heterogeneity, we try to characterize this complex by small angle X-ray scattering. And we use a tool that has been used for a long time. Indeed, oxyhemoglobin, which carries the heme hemicheiron in the ferrous form is known to form a complex with IsdB, but the heme structure is hampered. While methemoglobin, which is the natural substrate of IsdB, led the bacterium to extract the heme as well. So we collected the sex patterns for IsdB methemoglobin complex and IsdB oxymoglobin complex, and we first analyzed the one with the highest concentration possible, both to identify which species were present in solution and also to test whether interparticle interaction or aggregation were occurring. We found that at the highest concentration, the IsdB methemoglobin complex is formed by an hemoglobin dimer bound by two IsdB molecule, while the IsdB oxymoglobin complex was formed by an hemoglobin tetramer bound by two IsdB to the beta chain of hemoglobin. Then we also analyzed the entire concentration series of Sachs measurement, and we use oligomer to do the fitting. Indeed, this software allowed for the linear combination of multiple scattering pattern and allowed us to identify multiple species in each scattering pattern, and that led us to the conclusion that in solution, IsdB methemoglobin can dissociate to IsdB two IsdB molecule and the hemoglobin dimer, while the IsdB oxymoglobin complex will dissociate in hemoglobin dimer and a single IsdB bound. All this information were essential to proceed further, and indeed we go for static X-ray scattering at both small and wide angles. We collected indeed wide-angle X-ray scattering data at the highest protein concentration used by SAPS, since we defined that nor the aggregation or interparticle interaction were occurring, and also we employed the stoichiometry and the oligomeric species determined with SACS data, sorry. Moreover, these patterns that are shown here are the merged pattern of both SACS and WAX region, which increased the resolution a lot, since we have been able to go up to 1 angstrom to the -1 scattering angle, which is a resolution about 6 angstrom, spatial resolution of 6.3 angstrom. So since we still had a very strong data feed agreement, we could say that in the static X-ray scattering experiment, we have a dimer bound by two IsdB molecule for IsdB methemoglobin complex, and we will use it as a model afterwards, and a IsdB oxymoglobin, so a tetramer of hemoglobin bound by two IsdB and two exceeding hemophore in solution when we have IsdB oxymoglobin. These static experiments were essential as well for the fitting, as we will see. But now we'll just have to just look at how we develop our time-resolved wax setup. And indeed, we use the shutters and the chopper to select and X-ray pulses very, very small with a 20 microsecond dimension. And they were used to probe the reaction that was obtained by a stop flow apparatus, which mixes the two solution of hemoglobin and IsdB, and let the reaction started. and then we collect it using a CCD detector. The fastest way that we can obtain for the collection of data using the detector was a 30 millisecond acquisition. So to increase the time resolution, we acquired a sequence of experiment changing the timing of detection and irradiation, and then we just average the signal of the same time delay. And in this way, we increase a little bit more the temporal resolution. The result of this experiment can be visualized just by comparing the first and the last delay of the wax scattering pattern. And as we can see here, they are not completely superposed. So we have differences between the first time delay and the last one. So we can say that we are following some type of reaction. And if we want to see a little bit more how the data is describing the biological reaction, we can use the difference pattern, the difference scattering pattern. So the pattern that were obtained by subtracting all the scattering patterns at all time delay by the last one. So we can see a region where a big difference occur, which is at 0.0 angstrom to the -1 scattering angle, which is a resolution of like 80 angstrom. Then we have some difference here and here, which are at 0.5 angstrom to the -1, which correspond to a resolution of 13 angstrom. And then the last one with a resolution of about 10 angstrom. So these three region can help us to follow different conformational changes. The first one is more about change in stoichiometries or very big movement. The second one is about the movement of a small domain, and the third one was about the movement of small secondary structure in the protein. Another way to depict the time resolved wax patterns is this one, where all the pattern are shown as they are, so no different pattern but the direct one, but they are multiplied by the square of the scattering angle in a way that the information at higher angle, so the high resolution, spatial resolution is increased. To understand what we were observing, we feed the data as a linear combination of the static wax pattern that we collected before. We, of course, had the scattering pattern of IsdB and hemoglobin alone that I didn't show you before because they are just reference, but we use them here for the linear combination, and we use IsdB oxyhemoglobin as a model, so as a reference for the complex formed by an hemoglobin tetramer bound by two IsdB molecule, and then we will have the scattering pattern of IsdB bound to methemoglobin, which form an hemoglobin dimer, a complex formed by an hemoglobin dimer bound by two IsdB molecule. In the fitting, the fitting just found the best weight to multiply each single scattering pattern to improve the fitting of, to decrease the difference between the experimental and the predicted scattering pattern. Since we knew the concentration of the scattering pattern that we use as a reference, we obtain a concentration dependence of each scattering pattern along the time. And this is what we obtain. So we have at the first time delay that we acquired, the majority of the complex already formed. So the two IsdB bound to an hemoglobin dimer, which is depicted by the IsdB methemoglobin complex. And then we have IsdB, and then we have also a little of two IsdB bound to an hemoglobin tetramer. To further analyze this information, we employed SymBiology, which is a MATLAB tool that allow for modeling, simulating, and fit data about biological dynamic sample. It is mostly used for pharmacokinetics or metabolic pathway, but we adapted it to fit a kinetic profile as we add. We define the simplest and easiest kinetic model possible, where an hemoglobin tetramer, which is expected to be found in solution, is bound by two IsdB molecule, either at the same time or sequentially. And then we will have a dimerization that is irreversible, and in this case is essential to release by the steric clashes that is formed when hemoglobin is a tetramer, and then we will have a final formation of the complex. Using the fit data function of SymBiology, we've been able to fit our data and to extract the microscopic kinetic constants for all the steps that we define in our model. I just wanted to say that all the data are from the paper that we published, and it has the same name as my presentation. So if you want to look them again or at any time, you will find them as they are here. And just last information, we observed that we are not able to distinguish the heme extraction with the wax scattering pattern. So we decided to go and find complementary time-resolved spectroscopic approach. And as you might remember, we can follow the complex formation by fluorescence spectroscopy, and we can follow the heme extraction using visible spectroscopy. So we developed a setup using this similar stop flow apparatus, and we probed the reaction, either measuring the time resolved fluorescence signal with an excitation at 298 nanometer and an emission at 320 nanometer using a bandpass filter. And we also collected time-resolved optical absorption dataset via a photodiode array, and we processed the dataset, which is a series of visible spectra over time using SVD, which identify two main components. Here are the two kinetic profile of this component. So we try to couple them with the time-resolved experiment, time-resolved box experiment. But unfortunately, it was not that easy because visible spectroscopic data were not directly correlated to the concentration of the sample, since they depend on the extinction coefficient of the species in solution, or the emission of the chromophore. So we decided to use SymBiology to simulate the concentration profile of species in solution at the concentration that we use for a spectroscopic experiment, which by the way was the same as the time resolved box, so it is just a close call. But then we did a global fitting of all the data together using a sum of exponential function, which is a way to empirically follow the the biological process under investigation, and we found a good agreement and a very good fit of all the data, either the one from simulated using symbiology and the one from spectroscopy, so we can implement our kinetic model, adding at the final extraction step. All the steps are now described by observed kinetic constants, but they are related altogether. And what we found interesting is that the last kinetic observed constant is very close to the one reported for heme extraction by other author and by our previous work. To test further our methodology, we decided to simulate the concentration profile of all the species over time with a different concentration of protein. So we went for the mixture of 40 micromolar IsdB and 40 micromolar hemoglobin. We did the experiment, the spectroscopic experiment, again using this condition, and we did again the global fitting, this time using a 6 exponential model, and we still got a very nice fit, and we've been able to calculate the observed kinetic constant. What is interesting to observe is that, again, the last two kinetic constants are very close to the one that we calculated before, which told us that these two phenomena described by these two final exponent function are not related by the concentration, and therefore they are probably both related to heme extraction that occur after the complex formation. So with this experiment, we've been able to validate our initial kinetic model and also the microscopic rate constant that were obtained by the fitting of time-resolved wax. As a final step, we try to simulate the concentration profile of a protein in solution using a published paper as data from a published paper. In this case, the IsdB interaction with hemoglobin was followed just using spectroscopy and also using visible spectroscopy. They also use a different approach in the concentration, experimental concentration use. Indeed, they use just always the same amount of hemoglobin, and they increased the concentration of IsdB with the aim of working under pseudo first order condition. And all the data were just extracted from the signal at 406 nanometer, as well as we did in the first paper. They used a 4 exponential model. We did the same as before. We just simulated the concentration profile and under the experimental condition tested by the author, did a global fitting using a 5 exponential model. And what we found is that, interestingly, we've been able to simulate, observe kinetic constant that follow the same trend as the one obtained by the authors. Indeed, the first observed kinetic rate from the authors is consistent in some way with our data, and also the other one, the K2, is consistent with our data, while K3 and K4 still are in good agreement with our rate 4 and 5, which are related more to heme extraction, which are invariable to the concentration and more easy to simulate. So another time we've been able to test and validate our model and our kinetic constant that we obtained from time-resolved wax experiment. Just a couple of slides, more technical. The first one, I wanted to show that we also did IsdB analysis on the time-resolved wax data, which is more common way to analyze time result wax data, and we indeed identified three basis patterns, and those are the kinetic profile, which we still global fitted with multi-exponential model, which extracted three kinetic constants, which are in good agreement with one we obtain using the other approach, but are less informative, because if we wanted to start just by using IsdB, we would never be able to obtain the deepest understanding of our project, of our biological system. The very last one is about another region of the wide angle X-ray scattering, the one ranging from 1 to 2.2 angstrom to the -1 of the scattering angle, which is related to which is including the water scattering peak at 2 angstrom to the -1, and this is usually used to sense the local temperature of the sample when it is irradiated by the X-ray. In this case, the kinetic profile that is plotted in the second plot by correlating the intensity at 1.845 angstrom to the -1 and the simulated concentration of IsdB using our initial model show a good agreement in the first part. And since we know from isothermal saturation calorimetry that the IsdB binding to methemoglobin releases heat in solution, we can say that this part might be describing the complex, but we didn't analyze it further. It is just a way to increase the information that we can obtain from time result wax. Now, just a quick review of what we are still doing on IsdB hemoglobin complex. We obtained the cryo-EM structure of the pre-extraction complex between hemoglobin and IsdB, and it shows an hemoglobin tetramer bound by two IsdB molecule, which is in good agreement with our SOX and WAX did, which is cool. We also did screening to find molecule able to interfere with the complex formation between IsdB and human hemoglobin, and we found a promising molecule that we are now publishing after we have studied the Staphylococcus growth under condition where hemoglobin is the sole iron source, and we found that after 96 hours, when Staphylococcus aureus as hemoglobin can grow, while if we also add C35, which is the promising compound, it is not able to grow. So another good starting point for drug discovery point of view. Now I just have to thanks all the collaborators that work with me in this project, and in particular the Professor Luca Ronda and Matteo Levantino that are the corresponding author of the paper for all the analysis that we did together, and the funding for allowing us to do this project, and all of you for your attention.